I need to study A push though. Okay, anyways, I start recording. Okay.
Hello? Hello, welcome to this video.
Okay then, Brett.
Dare
Yep.
you share your screen with me?
Okay, fine. I'll go start sharing my screen with you.
Did you start the video yet or no?
Yeah, I already started.
Oh,
Uh,
am I in the video?
they're just
Am I
listening to us being retards right
am
now.
I gonna be famous?
Yeah, pretty much.
Mom, I'm on TV.
Oh my god, Brett. Okay, so most likely you're here because you don't know what the fuck was going on the entire year of bio now.
Hey, it's
And
half a year. Shut the fuck up, David.
oh, that's true. It's a half a year. Okay, and so here's a quick review of what we did over the year.
Oh, sound effects.
Yeah, thanks, buddy. Wow, look at all those people like reviewing. Lol. Oh my god. Anyways,
Oh, full screen.
yeah.
Wow,
And
there's
if
some
any of
shit
you don't know
happening.
who's in the background, that's Pratt's.
No, I'm sorry.
Yeah, yeah, there we go. Yeah. Okay.
Oh crap, I still needed the manual.
Oh, we had manual question. Oh yeah, we did have manual question.
Shit.
It's okay. I'm just gonna copy someone tomorrow.
Yeah, well, I have to turn it in tomorrow.
Oh, that's true. Well, that's awkward. Okay, so here's one of the questions we did in class, and it's the one about ecology. And I don't want to read this shit out loud though.
I got you. The following is a food web for a meadow habitat that occupies 25.6 square kilometers. The primary producer's biomass is uniformly distributed throughout the habitat and totals 1,500 kilograms per square kilometer. I'm not going to read that diagram, that's too much.
Yeah, okay. And then developers have approved a project that will permanently reduce the primary producer's biomass by 50% and remove all rabbits and deer. Which of the following is most likely to result as the completion of the project? So basically what they're saying is they're reducing the primary producers' biomass, which is the meadow grasses, by 50%, and they've killed off the rabbits and the deer.
Other one, yep, there you go.
Okay, so then what most likely would happen if that happened? It should be probably B, because like if you got rid of the rabbits and the deer, coyotes eat only deer and rabbits, so if you kill off both of those, the biomass of the coyotes should go down, because they don't have anything to eat anymore.
But David, aren't we allowed to make the assumption on C that they'll completely change their lives?
Sure, if you think coyotes can get voles, which are
The hell is a vole?
which is a type of bird. If you think they can like jump in the air and grab them, sure, yeah, we'll go with that.
Don't you know about or what's his name? The one from the Roadrunner?
Oh my god. God damn, French. Anyways, okay, next one. Another ecology question. The diagram above shows the progression of ecological events after a fire in a particular ecosystem. Based on the diagram, which of the following best explains why the oak trees are later replaced by the other trees? Okay, A. Eventually, the other trees grow taller than the oak trees and form a dense canopy that shades the understory. And that would be the right answer, actually, so I don't even need to look at the other ones, but we can go over on the other ones and explain why they're wrong. An oak tree alters the pH of the soil, making the soil of the forest better suited for shrubs and trees. Okay, why would the oak tree make the pH of the soil bad for its own environment? The oak tree wants to keep living. Why would it kill itself? That makes no sense. The roots of shrubs proliferate in the soil of the forest and prevent the oak trees from retaining water. Okay, so if the roots of the shrubs prevent oak trees from surviving, why are the other trees capable of surviving? That doesn't make sense either. And oak trees succumb to environmental pollutants more readily than do either the shrubs or the other trees. Okay, let's just hypothetically say that is true. That would still mean that there should just not be oak trees growing in the first place. It should just go from shrubs to other trees. No oak trees in the first place. So, yeah, the answer's A.
Wow, David.
Wow. I know, right, buddy? Okay, here's a question about DNA shit. I forgot what chapter this is called even.
Replication, chapter 16.
Oh, okay. Anyways, When DNA replicates, each strand of the original DNA molecule is used as a template for the synthesis of a second complementary strand, which the following figures most accurately illustrates an enzyme-mediated synthesis of new DNA at a replication point. Okay, first, what you should be able to recognize immediately is DNA replication always has to go to 3 to 5, so you can't have both of them going facing the same way, since DNA is anti-parallel, with one being 3 to 5 going one way and the other one going 3 to 5
It
the
goes
other
5
way.
to 3, not 3 to 5.
Oh shit, yeah, it is 5 to 3. Yeah, I was thinking in my head 5. I'm so bad.
Hey, it makes sense. I feel where you're coming from.
It's like, okay, yeah, it goes from 5 to 3. Okay, so you can immediately rule out A and C. And since it's being replicated from 5 to 3, it should be going this way. It should be D. Why is it D? Because this is 3 to 5 on the thing that it's replicating off of, so it's being read 3 to 5, but it's being replicated on a 5 to 3. And remember, 5 to 3 is a continuous. Going this way is always continuous, while the other one has to use Okazaki fragments, because how the replication fork works is it only can go like a certain distance, and then it has to jump back to the beginning over and over again. And here you go, and there's the rest. So it would go 5 to 3, 5 to 3, 5 to 3. Fun. What the hell is this? I don't know what that is. Cool, let's look over some of the things about ampicillin and that crap. I don't remember. Oh my god, it's such a big paragraph to read. Okay. In a transformation experiment, a sample of E. coli bacteria was a mix of a plasmid containing the gene for resistance to antibiotic ampicillin. Plasmid was not added to the second sample. Samples were plated on nutrient agar plates, some of which were supplemented with the antibiotic ampicillin. The results of E. coli growth were summarized below. The shaded area represents extensive growth of bacteria. Dots represent individual colonies of bacteria. Plates that have only ampicillin-resistant bacteria growing include which of the following? Okay. Obviously, you can just look at this picture and immediately rule out 1 and 3 if those are possible answers, because those have no ampicillin, so all types of bacteria should be growing, regardless. And then you can rule out the second one because it's completely blank, which implies nothing is growing on it. So the only answer is the one with 4, which is C. That's the same paragraph. The question is, which of the following best explains why there is no growth on the plate of 2? Okay, let's look through them. The initial E. coli culture was not ampicillin. Holy crap, what the hell's that in your background?
It's not me, I hear it too.
When you talk, it goes away. Oh, it's probably your mind. Yeah, some people, it's really weird. The initial E. coli culture was not ampicillin resistant. Yeah, that probably is it.
That's it? Yep.
Yep. Yeah,
Can
alright.
confirm.
I can just go for the other ones and show why those are wrong. The transformation procedure killed the bacteria. Well, if the transformation procedure killed the bacteria, there shouldn't be growth on the 4th one either, because that was also a transformation procedure. So neither of these plates should have anything growing on them. So that's wrong.
That's not the same procedure, David.
What?
It's not the same procedure. Try again.
What? Pratt,
the procedure is going horizontally, not vertically.
Pratt, what?
You gotta explain correctly,
Oh,
David.
shoot, right, yeah. I'm dumb as shit.
God damn it, David.
God damn it.
One job.
I know, right? Okay, then why is it wrong?
Oh, because if it was just the transformation killing the bacteria, there could still be other bacteria growing there.
Okay. Seems legit. Nutrient agar inhibits E. coli growth. Okay, all plates should be empty if the nutrient agar is inhibiting the E. coli growth. And the bacteria on the plate were transformed. Okay, if you know anything about what the transformation is about, it's supposed to make them resistant. So if they were transformed, there should be growth. So that's like the exact opposite of what the question is asking for. So that's probably not the answer. So yeah, it's A. Plate 1 and 3 are included in the experimental design in order to... One, Pratt, please.
I'm telling you, it's not me. I hear it too.
But do you notice it goes away once you talk?
Oh yeah, shut up. We're done now.
Yeah, it's like not there anymore. But then afterwards, like if you stop talking for like 10 seconds, it like comes back in. It's really...
There's literally no background noise in my room.
But it's not your back. It's like your
Okay,
mic.
wait,
It's
hold
like
on. I'm gonna be quiet and monitor my mic levels. Just keep going.
Okay. 1 and 3. Demonstrate the... What was the purpose of 1 and 3? Demonstrate that E. coli cultures were viable. Yeah, pretty much. That should be the answer. These demonstrate that the E. coli can grow on that, and then not like the E. coli is already dead. And then why is it not B, C, and D? Demonstrate that plasmids can lose its ampicillin-resistant gene. Okay? This is talking about not putting ampicillin onto them. So how could you even test if the ampicillin gene was lost if there's no ampicillin in the first place to test it with? Demonstrate that the plasmid is needed for E. coli growth. No, because we just proved, as you see, the wild type without the plasmid, it's growing. Without the plasmid. And prepare the E. coli for transformation. No, how does this have anything to do with the transformation on the other half? Like, this is like a completely different petri dish. It's not like one half is one side and one half is the other. Okay then, so that's why it's A. Oh my god, there's more of these questions? God damn. Which of the following statements best explains why there are fewer colonies on plate 4 than plate 3? Okay. Plate 4 is the positive control. No, control implies nothing's been changed, so it can't be the control, because that had ampicillin and the plasmid was added. Not all E. coli cells are successfully transformed. Yep, that's the answer. C. The bacteria on the plate 3 did not mutate. What?
Not sure. Well,
What?
not sure what that would mean.
I mean, some of them might have not mutated, that's true, but some of them probably did.
But
like,
would it matter?
Yeah, it really doesn't. So that really doesn't matter about C. And the plasmid inhibits E. coli growth. No, because the plasmid obviously allows it to grow where there's ampicillin, so it's not inhibiting growth. Okay, so yeah, it would be B. Holy crap, how many more? Okay, there's one more. In the second experiment, the plasmid contained the gene for human insulin as well as ampicillin-resistant gene. Which of the following plates would have the highest percentage of bacteria that are expected to produce insulin? Okay, it would not be the number 1, because this implies that you never put the ampicillin-resistant thing into it, which means there's no insulin. So it would be 0% would create insulin, which would be a very bad percentage of insulin production. Number B is free. That would be a better percentage than the first one, but based on the earlier question that we did, as you saw, there's only a certain percentage of them is actually transformed. Not all of them are transformed. So you might get maybe, based on trying to get a percentage of this over that, maybe 5% insulin production. That's still pretty bad. And then 4, yeah, 4 is the best one, because that would be 100% insulin production, because the only things that are left are the ampicillin resistant ones that also have insulin, which means it's 100% insulin production. And then D is not correct, because 1 and 3, 1 has 0% and 3 has 5%, both of which are very bad. So it's C. Oh, look, more questions. A simple cuboidal epithelial cell line the ducts of certain human Hey, I remember that from Otto's anatomy. Anyways, various materials are transported into or out of the cells by diffusion. The formula for surface area of a cube is blah. Formula for the volume of a cube is blah, where S is the length of the side of the cube. Which of the following shade?
Profound.
What?
Profound.
Okay. Which of the following cube-shaped cells would be most efficient in removing the waste by diffusion? Okay, so basically you want the greatest surface area to volume ratio. And surface area to volume ratios are best when the cube is the smallest, so it's A.
If it comes to worst, you can literally just plug it in, solve for it.
Pretty much. You could find the surface area of A over the volume of A, and then do the... You don't need to test the two middle ones, because you know it has to be one of the extremes, so that you can test D and find the surface area of the large one divided by the volume of the large one. And you realize the smaller one has a way bigger surface area to volume ratio. Sickle cell anemia results from a point mutation in the HBB gene. The mutation results in the replacement of an amino acid that has a hydrophilic R group with an amino acid that has a hydrophobic R group. Hydrophilic R group with amino acid that has a hydrophobic R group on the exterior of the human globin molecule. Such a mutation would most likely result in altered properties of the molecule as a result of abnormal interactions between adjacent hemoglobin molecules.
Yes, that one.
Possibly, I'm not 100% sure.
No, I mean, I just did this problem. It's that one.
Oh, okay. Alright. DNA structure as a result of abnormal hydrogen bond. No, because remember that it says amino acid. This is a protein. It has nothing to do with DNA. Fatty acid, immediately can rule out that one as well, because once again, this is amino acids, and amino acids make proteins, not fatty acid structures. A protein secondary structure as a result of abnormal hydrophobic interactions between R groups in the backbone of the protein. Why is this wrong?
Because R groups are tertiary, not secondary.
Yeah, there we go. Okay then. So it's A.
I think that was it. I'm pretty sure that was the last one.
Yeah, but people usually... Because remember that 2/3 of the test are actually the actual slides, so...
True.
Usually on these things I usually review all the slides. Oh, great.
Alright, David, I'll help you out.
No, I can do them. It's fine. It's just like, what the fuck. Okay. Cool. Talk about mutations. Point mutations can affect protein structure and function. Okay, so a mutation can cause a change in how it looks and whatnot. That's really surprising. Mutations are changes in the genetic material of a cell or a virus. Yep, makes sense. These include large-scale mutations which along segments of DNA are affected. Example, translocations, duplications, and inversions. So basically what they're talking about in that case is basically mutations happen when the DNA is altered in somehow. And translocations, duplication, and inversions are all types of alterations to the DNA. And a chemical change in just one base pair of a gene can cause a point mutation. So basically, think of a point mutation like on a line, it causes one point of the line to be changed. Instead of like the entire line being changed, just one point of the line is being changed.
It'd be helpful if you had a drawing.
But that requires me now to like open a freaking like
Or just go to the next slide. Or go to the next slide.
What do you mean on the next... Wait, was there a drawing on the next slide?
Yes.
Oh, well, that's awkward. See, I don't look at these slides ahead of time, so I would not... Oh, yeah, there is. Oh. Okay. You're cool, Prats. For example, sickle cell disease causes by mutation of a single base pair in the gene that codes for one of the polypeptides hemoglobin, which we just did in that multiple choice, and so a change in the single nucleotide from T to A in the DNA template leads to a normal protein. So, basically, this is what hemoglobin should look like. In a point mutation, just one area, which is this middle one, is changed from a T to an A. And now you have, instead of getting, I don't remember what GLU stands for, you get valine. A point mutation results in a replacement of a pair of complementary nucleotides with another nucleotide pair is called a base pair substitution. Okay, so basically, remember that in DNA, when you replace one of these, it has to have the other one change as well, because you can't like have an A with, you can't have an A with an A, they don't bond together, you know, both of them will need to be replaced. Plus, you get an entire base pair. substitution. Some base pair substitutions have little or no impact on the protein function. Okay, cool. In a silent mutation, alterations in the nucleotides still indicate the same amino acids because of the redundancy in the genetic code. And other changes lead to switches from one amino acid to another with similar properties, and still other mutations may occur in a region where the exact amino acid sequence is not essential for function. Okay, is there a diagram of that shit? No, that's awkward. Now I'll just go over which one each of these are. So basically, remember that in that diagram of mRNA.
Oh, it's like 3 slides down.
Oh, that's okay.
Or 5 slides down. 29.
Oh my god.
Right, no, you missed it. Go back. There.
Oh no, but that's talking about Frameshift. I'm
Wait,
talking
what are we
about,
on?
you know that table that converts the mRNA into like amino
Oh, a
acids?
codon table?
Yeah. I was just going to
Oh.
look up a codon table. Doesn't look like she has it anywhere nearby.
Yeah. Yeah, you're gonna have to look that up.
So basically, a silent mutation is basically...
Yeah, I'm not seeing one.
Okay, so here's a codon table. Basically, a silent mutation. Notice how UUU makes this and UUC makes this. So if you replace this U with a C, it doesn't really change anything because it still makes the same amino acids. So it's silent. It has no actual change. And then we have leads to other changes lead to switch from one amino acid to another with similar properties. Okay, remember that a lot of these ones are some of them are hydrophobic, some of them are hydrophilic. If you replace a hydrophobic one with a hydrophobic one, it usually won't have that much change. Because they're hydrophobic, they're both going to dislike water, so they both kind of have similar properties and despise water. And then other changes lead to switch from one amino acid type to another. Still other mutations occur in a region where the exact amino acid sequence is not essential for the function, so... Oh my god, that's not the protein I was like... Proteins...
What the fuck is?
That wasn't the protein I was looking for.
David, I looked up from my Twitter for 1 second, and that's all I see, you weirdo.
Okay. Okay, so here's like a protein structure. Anyways, basically, imagine if the protein structure is like all the way at the middle of this protein. If it gets altered, it's not going to have that much effect because what's being changed is probably going to be near the outside, not the interior of the protein, so it doesn't matter if the interior gets altered. Because all that matters basically for a protein is that it's... Crap, what is that area that the actual... like
Active site?
Yeah, there you go. The active site is changed. All the other part is the structure. If the structure changes, it really doesn't matter. And then other base pair substitutions can cause a readily detectable change in the protein. These are usually detrimental, but can occasionally lead to improved proteins or one with novel capabilities. Changes in amino acids at crucial sites, especially active sites, are likely to impact function. Missense mutation are those that still code for amino acid that change the indicated amino acid, and nonsense mutations. Press, please. Talk.
Dude, that's not me. I'm just monitoring my mic levels. It's not changing.
But it goes away when you talk.
Dude, oh, fucking, I don't know. It's somewhere along the way, then.
Yeah, I don't know where it's coming from either. Nonsense mutations change the amino acid codon into a stop codon, nearly always leading to a non-functional protein. Okay, so missense. Just kind of think of it as like it just spews out random shit. That missense mutation spits out random shit, and a nonsense mutation completely stops it. It should make more amino acids, but since there's a stop codon, it stops making amino acids. And here is a diagram of it. So here's a silent mutation, has no effect, as you can see. It still makes the correct thing. Here is a missense one. Basically, it actually changes, so instead of being GLY, it turns into SER. And then a nonsense one, as you can see, it actually stops it immediately, and now you only have the methodiene. Is it called methodiene? Whatever. You only have the met amino acid. You don't have any of the amino acids. Insertions and deletions are addition or loss of nucleotide base pairs in a gene. They have disastrous effects on the resulting protein more often than substitutions do, unless these mutations occur in multiple free. They can cause a frameshift mutation. All nucleotides downstream of the deletion or insertions will be improperly grouped into codons. The result will be extensively missense, ending sooner or later in nonsense, premature termination. So yeah, basically, when you insert something, note how it's always in groups of three. If you add something, everything's going to be shifted over one, so everything's going to be all out of whack if that happens. You can see a frameshift causing a missense. You added a U, the U got deleted from it, and now, instead of making this, it makes these things. It makes LEU and LAA. Everything gets fucked up. Frameshift causing immediate nonsense. So basically, the U gets put in, and what should be AAG for LYS, it makes a UAA, and UAA is a type of stop codon, so immediately stops. And insertion or deletion of free nucleotides, no frameshift, extra or missing amino acids. So basically, if you pulled out 3 of them, basically there should be a LYS, but now there's no LYS. There's just one amino acid missing now. Mutations can occur in a number of ways. Errors can occur during DNA replication, DNA repair, or DNA recombination. Yep. These can lead to base pair substitution insertions or deletions, as well as mutations affecting longer stretches of DNA. These are called spontaneous mutations. So basically, these are the type of mutations that happen over time, like entropy. No, let's not talk about entropy. Just know that over time, as things get older, things get fucked up, and these kinds of errors are bound to eventually happen. Thus, the spontaneous. Mutagens are chemically or physical agents that interact with DNA to cause mutations. Physical agents, including high energy radiation like X-rays and ultraviolet light, can cause these. Okay, so if you want to just think about something kind of relevant, not really, you could think of the X-Men as sort of a mutagen. That's basically, they get exposed to, in the comics, they get exposed to mutations and they have extra abilities. Yeah, that's a really bad example, but basically when you get exposed to higher energy radiation, you can have DNA mutations. Chemical mutagens can operate in several ways. Some chemicals are base analogies that may be substituted into DNA, but the pair incorrectly during DNA replication, or mutagens interfere with DNA replication by inserting into DNA and distorting the double helix. Still others cause chemical changes in the bases that change their pairing properties. Okay, basically, there's multiple ways that chemicals can cause mutations. You really don't need to probably know the specifics of how they do it. Like, example might be benzene. Like, benzene is like this very stable molecule within, or chem, if you expose DNA to that, it'll just like bind with everything and just fuck everything over.
Yes, the phenyl groups.
Yes, they just fuck everything over. Let's look at the mutate. Nope. Okay. How do genes move? Examples of when genetic material jumps from one location to another. They have translocations, duplications, and crossing over. Brad's talk. Okay, it went away again. Thanks, Bryce.
I'm trying to let you do your video, man.
Yeah, but it's like the thing keeps like accomplished
Kill
when
yourself,
transposase enzyme acts on transposon DNA segment that jumps. The transposase enzyme recognizes inverted repeats at the edges of the transposon. Transposase cuts the transposon from its initial site and inserts it into the target site. The simplest bacterial transposon and insertion sequence consists of only the transposase gene. So basically, what the hell are they talking about?
Did it go away?
What?
The sound? Like when I wasn't talking?
What? What?
Wait, it goes away when you talk too.
Yeah, I think it's something to do with Skype itself. I don't really know if it's like the call. It's like Skype is just a derp as hell.
True.
So basically, what the hell is going on? They basically are saying this is the transposon on its initial site, then there's transposase that cuts it out and then shoves it inside. And now it's the transposon at the new site. Yep. That's pretty much what it does. It's not really much to talk about for that. Transposose is a piece of DNA that can move from one location to another in a cell's genome. Okay, so transposose is a piece of DNA that can go from one area to another. Transposose movement occurs as a type of recombination between transposose and another DNA site, a target site. In bacteria, the target site may be within the chromosome, from a plasmid to a chromosome, or vice versa, or between plasmids. Transposons can bring multiple copies for antibiotic resistance into a single R plasmid by moving genes to that location from different plasmids. This is why some R plasmids convey resistance to many antibiotics. Composite transposose, complex transposons, include extra genes sandwiched between two inserted sequences. It is as though two inserted sequences happen to land relatively close together and now travel together, along with the DNA between them, whereas a single transposons. Okay, so basically, complex transposons, it's just basically a longer transposose, so now it can do more than one function because it has two genes inside of it instead of one gene, or three genes instead of one gene. It's just a longer transposose that does more things. Because you see now this one has three of them, and it has antibiotic resistant, and it has a different transposose, and it has an initial transposose, so it has multiple transpososes now. While certain sequences may not be beneficial bacteria in any specific way, composite transposons may help bacteria adapt to new environments. For example, repeated movements of resistant genes by composite transposons may concentrate several genes for antibiotic resistance onto a single R plasmid. In an antibiotic-rich environment, natural selection favors bacterial colonies that have built up composition R plasmids for a series of transpositions. So basically, holding multiple plasmids within the cell is not efficient. You don't want tons of plasmids. You just want like one or two. So basically, all you do is like you throw in maybe like all the ones that you need onto one plasmid. So now you only need to make one plasmid per cell instead of like 200 plasmids per cell, thereby wasting less like resources into making more plasmids. Ooh, viruses. Evolved from plasmid transposons most likely. Mobile genetic material, more closely related to the host cell DNA than other virus DNA. Crat's off.
Your video sucks.
I agree, it does suck. Why am I making them? Probably enter damaged cells first, then once developed, capsid envelope could enter undamaged cells also. Now, ATP curriculum focuses on how they can introduce genetic variation. So here's a diagram of like the virus compared to the bacteria. Now how the virus is way smaller than a bacteria, and then the bacteria is even way smaller than an animal cell. And here's different types of bacteria that there can be, and there's a lot of them, so I'm not even going to go over all of them. Capsid is a protein shell that encloses the viral gene. Okay, so basically, think capsule. Capsid, capsule. A capsule is something that carries something. So the DNA is inside the capsid, or the capsule in this case. Capsids are built of a large number of protein subunits called capsomeres, but with limited diversity. So each one of these things that makes up the capsid, is made of capsomeres. The capsid of the tobacco mosaic virus has over 100 copies of the same protein. Adenosine virus have 252 identical proteins arranged into a polyhedral capsid as an isohedral. Okay, they make types of shapes. Basically, they make uniform shapes. Congratulations, wow. Some viruses have viral envelopes, membranes, coatings, their capsids. Basically, these ones are one up from the other ones. Before, there's just this protein thing on the outside. Now they also put membrane on the outside. These envelopes are derived from the membranes of the host cell. They have some viral proteins and glycoproteins. So they have some viral proteins on the surface and some glycoproteins. Yeah. The most complex capsids are found in viruses that infect bacteria, called bacteriophages or phages. The T11 phages that infect E. coli have 20 side capsid heads that enclose their DNA and a protein tail piece that attaches the phage to the host and injects the phage DNA inside. So basically, this is its shape. It's kind of like this interesting, kind of like a spider shape sort of thing. It's like there's its legs that attach it to it, and then here's the actual tube that then injects its DNA that's stored up here into the cell. Can viruses, living or not, can reproduce only within the host cell. An isolated virus is unable to reproduce or do anything else except infect an appropriate host. Host range, lock and key, fit between the proteins on the outside of the virus and specific receptor molecules on the host cell surface. Some viruses, like the rabies virus, have a broad enough host range to infect several species, while others infect only a single species. Most viruses are specific tissues. Please.
Bruh, don't hate on me.
Oh my god, Prats, I'm sorry. Viruses take over the machinery of the host cell. Okay, basically, in this case, by most definitions, viruses are not considered living by most biologists as of currently, basically because they don't fit the criteria of living, because living usually means they can react to, they attempt to maintain homeostasis throughout their time, but a virus, when it's outside of a host cell, makes no attempt at maintaining homeostasis, just accepts what the environment is like outside. And here is the virus taking over cells. Yay! Virulent phages and the Lytic cycle. Okay,
Do we
so
know?
what?
I thought she said we didn't need to know Lytic and what's the other one?
She did?
Thought, like, right?
I thought.
It's like Lytic and
If we don't need to know, Lytic cycle and lysogenic, I'll just like go over very quickly. So Lytic cycle is basically, it immediately puts in its mRNA, DNA, whatever it is. It goes in, takes over the cell machinery, starts making duplications of itself. Then once it's done, all of them rupture out, killing the cell and making tons of viruses. Phages have the potential to wipe out a bacterial colony in just hours. Bacteria have defenses against phages. Natural selection phases, bacteria mutations with receptor sites that are no longer recognized by a particular type of phage. Bacteria produce restriction nuclease that recognize and cut off foreign DNA, including certain phage DNA. Modifications to bacteria's own DNA prevents its destruction by restriction nuclease, but natural selection favors resistant phage mutations. Okay, and here are the diagrams of the two different types of... Prats, what the hell?
I'm watching a Vine.
Okay, here's the two different types. Lytic cycle, as you see, immediately gets inserted, immediately starts doing its thing, makes new phages, phages release from the cell, killing it. Then it goes on the process trying to find a new one to do it over again. So this is the lysogenic cycle. Note how it's a little bit different. So, as you can see, it does get injected. But when it gets injected, it integrates itself into the actual DNA. Kind of just stays there. And as the cell reproduces, it makes more copies of this DNA. And it just kind of stays around for a while. For a long time. As the cell keeps making more of themselves, eventually it enters a Lytic cycle. And once it enters a Lytic cycle, it does the same thing as the original Lytic cycle did. Makes more copies, bursts from the cell, killing the cell, and making more of them. So basically, this is kind of like a more stealthy thing. It waits around, lets the cell make like hundreds of copies of itself before it then ruptures out. So it can like spread its disease everywhere. Instead of just having one cell make just like 100 copies, you can have like 200 cells making 100 copies. Thereby making you have like a 200-fold increase of viruses being released. Here's retrovirus, which is HIV is a type of retrovirus.
We didn't cover retroviruses, like.
What?
So, or no, we did, but it's not on there for sure.
Oh, okay. Never mind. Why do we have slides on retrovirus
Like,
again?
it's like, you should know it, but they're not testing on it specifically.
Okay. Skipping that then.
Or I think that's what she said.
Okay,
So,
I'll just go over slightly then. Okay.
yeah.
Retrovirus, basically, what's interesting about a retrovirus is it has RNA inside of it, but it has something called reverse transcriptase, which is a type of enzyme that converts RNA into DNA. It's called reverse because normally DNA goes into RNA, as most cases, but with reverse transcriptase, they can convert RNA into... Holy shit, bro.
Sorry.
Convert it into DNA, and then the DNA can integrate itself into the nucleus of the actual host organism, which in this case are our T white blood cells, which is pretty bad because it kills off all our white blood cells, and then we're pretty susceptible to all types of diseases. But anyways, vaccines. Okay, vaccines basically, they allow our body to prevent viral infection because they already see they already know that it's a virus. Your body mounts a defense against it. The virus never can do anything bad to us. And then remember, antibiotics only work on bacteria because viruses aren't like bacteria in the fact that bacteria are actual living things, so you can just mess up their homeostasis and then they'll die. But a virus, you can't mess up homeostasis in a virus because they don't care about homeostasis. Antivirals interfere with viral nucleic acid synthesis. AZT interferes with reverse transcriptase of HIV, and whatever that inhibits herpes virus DNA synthesis. So basically, antivirals, they interfere with this process of converting the reverse transcriptase into this DNA, thereby preventing it from actually doing its job. So the virus stops working, basically. Emergence of new viral diseases. Okay. And what are they caused by? Mutations of existing viruses. Don't even really want to read that, but basically, when it mutates, most of the time mutation will probably be bad, but sometimes it might make it better, like might be more resistant to antiviral medications, so more likely to survive and keep reproducing. Spread of existing viruses from one host species to another. Wow, I hear the sirens in the background there, perhaps.
Did you hear the love yourself?
Okay, then. So basically, sometimes the virus can jump over from one species to another. So, as this case, the hantavirus went from mice to people. Spreads from small isolated populations to widespread epidemics, like AIDS. Note that AIDS used to be only in Africa, and now AIDS is everywhere. Viral disease in small isolated populations can emerge and become global. New viral diseases can emerge when viruses spread from animals to humans. Viral strains of jump species can exchange genetic information with other viruses to make humans have no immunity. And these strains can cause global epidemics. 2009 flu pandemic was likely passed to humans from pigs. For this reason, it was originally called the swine flu. Okay, yeah, that's just kind of some interesting terminology. Basically, a pandemic is one area, a major region. No. Epidemics are a major region has an infection. So, like, you can... Crash, please. Epidemics can just have a major disease in one region, but a pandemic has a major disease across the world. Oh, cool. Review questions now. The chemical reaction for photosynthesis is... this really long ass thing. Yeah, fun. The input of water is labeled radioactive isotope oxygen, then the oxygen releases as the reaction proceeds is also labeled, which the following is most likely explanation. Okay. During the light reactions of photosynthesis, water is split, the hydrogen atoms combine with the CO2, and the oxygen gas is released. Okay. I actually have no I totally don't remember how to do these kinds of things. Let me just read through all the answers. During the light reaction of photosynthesis, water is split, removes electrons of proteins, and oxygen gas released. During the Calvin cycle, water is split. No. The ones of it has nothing to do with the Calvin cycle, so you can already remove C and D. So it has to be A or B. Water is split, the hydrogen atom combines with CO2. No, Calvin cycle is the one that deals with CO2, so it has to be B, because Calvin cycle deals with CO2, so this should have nothing to do with CO2, so it can't be A, so it has to be B. During the Lytic reaction of photosynthesis, water is split, removing electrons and proteins, and oxygen gas is released. Yeah, now that I remember. The light is used to knock off electrons, which are then used to deal with the... Oh, crap. What is
The
that
light?
thing that makes the gradient of hydrogen ions? What is that enzyme thing called?
Oh, ATP synthase?
Yeah, there we go. Yeah, the light reaction is used to knock off electrons, which are eventually used for ATP synthase purposes. So, it's B. Ooh, this one. Oh my god, but there's such a long ass.
I thought we already did this one.
But we didn't do that in the video.
Oh, you're right.
Yeah. Paramecia are unicellular protists that have a contractile vacuole to remove excess intracellular water. An experimental investigation, paramecium was placing cell solutions of increasing osmolarity. The rate at which the contractile vacuole contracted to pump out excess water was determined plotted against osmolarity of the solution as shown in the graph. Which of the following is the correct explanation for the doubt? At high osmolarity, lower rates of contraction are required because more salt diffuses into the paramecium. No, that's not it. Because it's not the salt that's diffusing across into the paramecium, it's the water that's diffusing into the paramecium. The contractile rate increases as osmolarity decreases.
It's B.
Because the amount of water into the paramecium by osmosis increases. Yeah, it is B.
You're welcome.
The contractile vacuole is less efficient in solutions of high osmolarity because of the reduced amount of ATP. That's just random bullshit that they hope makes sense to dumbass people. That's just not seen. They just threw random shit together and then they call that an answer.
You went to hell for saying that.
What?
You went to hell for saying that.
It's unfortunate. I was already going to go to hell anyway, so it really doesn't matter.
True.
In osmotic salt solution, there is no diffusion of water into or out of the paramecium, so Okay, that's actually somewhat true. But it's kind of wrong. So it's not that there's no diffusion. The diffusion rates are equal, so the amount of water going in and out is equal. So that's kind of wrong, but it's sort of true. Yeah, it's not that though. Anyways, it's B. Oh my god. No, it's mutation practice. We'll do it. How many more slides are there? Holy crap, how many slides are there? Holy shit, there's like 100 more slides.
Good
Fuck
luck, David.
this shit. I'm not making... I'll just make it like... Whoever has bio tomorrow sucks to be you. I'm going to make a part 2 later.
Hey, that's me. Fuck you, David.
Prats, do you really need help?
Not really, but
Yeah, exactly.
A lot of these slides are actually just the same thing, just in more detail.
Well, fuck, I'm not going through that shit right now.
So, like, the one you're on right now, it's like all the steps of binary fission are behind it.
Yeah, well, fuck. Okay, I'll make a part 2 probably on tomorrow. Yeah, probably make a part 2 tomorrow. Anyways, bye guys.
Bye.